Okay, welcome back, guys. We got some more content to cover today regarding muscular system. So let's go ahead and continue moving forward. So let's see where we're at. When we were last talking about this, the last video was covering quite a bit of content. But now we get to talk a little bit more along the lines of how the muscle contracts itself. So that's really what we're going to be focusing on. So we were previously looking at the microscopic things, especially like the muscle cell itself, what we see here, or a muscle fiber as we call it. Then we zoomed in and looked at the myofibril. Now we get to zoom in a little bit further and look at the sarcomere. So, first of all, what is a sarcomere? So, a sarcomere is considered the contractile unit of a muscle fiber. So, if I were to say, like, what is the unit of the muscular system? A sarcomere. It's the building block. You need sarcomeres in order to build a more complex structure, such as a muscle, right? Like an actual organ. Here's what the sarcomere looks like. This is just a rendition. There's a lot of different renditions that we have, right? And by the way, not a bad idea to note this. The sarcomere, as you can see here at the bottom, is an IAI repetition. I just forgot to write this earlier. So not a bad idea to be aware that it's just an IAI repetition. I band, A band, I band. If you recall from our previous video, the idea behind that is the I band is the light region and the A band is the dark region. So that's what we observe. And it has to do with the proteins that are found there. So that's what we'll discuss. So there are two major proteins found in the sarcomere. We have a thick filament, which literally is just a thicker protein, and we call that protein myosin. There is some other stuff here too. We have ATPase. Anything ending in ase is an enzyme. Enzymes are proteins that help to accelerate the reaction, which we've just talked about that previously. But ATP is your energy currency. It's like the dollar for the US economy, right? So ATP is the currency of energy. ATPase helps to break down ATP and release said energy. The analogy I like to give is like in the context of like dollars, it's as if you're bringing a dollar to like a gas station and you're buying something. So you're exchanging your currency for a product. And that's kind of what's happening here. So if that's the thick filament, we also have the thin filament. The thin filament is going to be actin. So for our intents and purposes, honestly, the big thing here is just pay attention to what is what, like which is which. Can you identify that the thick filament is going to be myosin and the thin filament is going to be actin? I do have a mini mnemonic here that I think helps. It helps me out. That's what mnemonics are for, memory purposes, right? When I hear actin, I think of the Atkins diet. The Atkins diet is rather popular. It's been popular for a long time. It hasn't necessarily been as popular in recent times, but the Atkins diet is a diet for losing weight and becoming thinner. So actin and Atkins sound very similar. So I think, okay, Atkins is a diet for losing weight and becoming thinner. Actin is the thin protein or the thin filament. So as I mentioned, we're trying to learn more about how the muscles contract. But before we get to that, there are some things that we need to discuss. So first of all, regarding the thick filament myosin. Myosin filaments are going to have their own heads or cross bridges that we see here. These are things that extend outward and attach themselves to actin or the thin filaments. Myosin and actin do overlap a little bit, but not so much. And we even have a region here called the bare zone, which is just an entire area that there is no protein. Now, the stuff we're about to discuss is more oriented around nervous system. Now, the nervous system is the next unit we're going to cover, so it's all related. It's tangentially related. So this is stuff that we're going to see again in nervous system anyway, so we might as well talk about it now. There are two properties of skeletal muscles that we need to discuss: irritability and contractility. Irritability is the ability to receive and respond to a stimulus. So this is the ability for your body to receive the stimulus. And where are these stimuluses or stimuli coming from? The brain. The brain generates this electrical current. Contractility is the ability to shorten when the stimulus has been received. So why is this important? Let's say you get admitted to the hospital. You're admitted to the hospital and you need medical attention. And the reason why you need medical attention is like your arm isn't working. You can't bend your arm. Okay, well, they have to diagnose you and run some tests and figure out what's going on. If they can observe that it's an irritability problem, then they could identify, okay, you're not responding and receiving signals properly. So that could be a nervous system issue. That could be a brain problem, a spinal cord problem, nerves. There's a lot of issues there that could be pretty catastrophic. Contractility means that the nervous system is functioning. You're sending the signals properly, but the muscle itself is not contracting properly. So that could be a muscle issue, which could be better treated. So as I mentioned, this is stuff that's a little bit more oriented around. I didn't even notice this typo here. Some stuff oriented around nervous system. The big thing here to recognize is that every skeletal muscle needs a nervous impulse. If it doesn't have a nervous impulse, it's not going to activate. The rest of this stuff, motor units, neurons, that's stuff from nervous system that we will learn in future videos. Now, nerves are able to communicate with each other exceptionally. They use electrical currents, action potential. This is stuff that we're going to learn in our future videos, but nerves have a harder time communicating with non-nervous tissue. So they have to find a way to do that type of communication. So what do they use? They use junctions. In this particular case, we have what's called a neuromuscular junction. Simply enough, it's a connection between the nervous system, like neuro, and the muscular system, literally muscles. So it's just an association site. There is some stuff happening here that we need to discuss. So let's get into that a little bit. If you had a powerful microscope and you were watching or observing a muscle and you were seeing it, it would look like the muscle and nerve are touching. But if you had a powerful enough microscope, like really, really powerful, you could actually see, and you can see in this picture here, there's a very small gap between the nerve and the muscle. And we call that the synaptic cleft. The synaptic cleft is this gap between the nerve and muscle. The nerve and muscle never physically touch. So that's something you may want to really pay attention to just in case you see that for a future question. What do we find in that area? Interstitial fluid. It's the magic stuff that's found all over the place as far as in between fluid. Okay. How do these neurotransmitters, or how exactly do the nerves communicate with the muscles? Well, they don't use an electrical impulse, they use neurotransmitters. So, neurotransmitters are these chemical messengers. That's the big thing here. They are these chemical messengers that are released. And most of us have heard of neurotransmitters before, even if you've never seen this word, because a lot of neurotransmitters are rather common in pop culture, dopamine being a big one, serotonin, endorphins, right? The one that we really need to pay attention to for this unit is acetylcholine. Acetylcholine is the one we really want to pay attention to. And the reason for that is because acetylcholine is the neurotransmitter responsible for skeletal muscles to activate. So we really want to pay attention to that one. The rest of the stuff is more oriented to the nervous system, but we might as well talk about it. The neurotransmitter will attach itself to receptors of the sarcolemma, which is the plasma membrane of the muscle fiber. The sarcolemma becomes permeable to sodium. Permeable meaning it allows for these ions to flow through. Imagine like a gate opening up. Sodium will rush into the cell, generating an action potential, which we'll get into that in nervous system. Once started, muscle contraction cannot be stopped. That is actually important, and we'll talk about that again before this video ends. Finally, here's the slatting filament theory in like written format and step by step, right? There's technically 4 steps if you want to think of it this way. Step 1 is always you need a nervous system impulse. You need a nervous stimulus. So the nerve has to activate the myosin, and the myosin will attach itself, its cross bridge as it's called, to attach to the actin. Step 2, the myosin is going to attach to the actin and then drag itself to new sites, to the next site on the actin. So it's already starting the slatting process. Step 3, the myosin literally just continues to slide across the actin. And then step 4 is rather self-explanatory. But once all of this has happened, the result is the muscle is shortened. You've successfully contracted the muscle. And then, of course, after that, what's going to happen? It's going to relax and it's going to expand. So it's a contraction and relaxing. What's really fascinating about slatting filament is that this has to happen within hundreds of thousands of different sarcomeres that make up your muscles. Now, the question is, do all of these sarcomeres and all of these muscle fibers activate simultaneously? Not exactly. So that's what we're about to discuss. Here we can see in 2D form what's going on with slatting filament theory as far as the myosin attaching to actin, the myosin heads moving across the actin and dragging everything in. All right, so not too much left. Muscle fiber contraction is all or nothing. That's important to understand. So once it starts, it's going to happen, right? But these two bullets here actually make an interesting point. Within a skeletal muscle, not all fibers may be stimulated during the same interval. Different combinations of muscle fiber contractions may give differing responses. So what does that mean exactly? What that means is that every fiber that we have, and we have, again, thousands upon thousands of fibers that make up your muscles, not every fiber needs to activate. Not every fiber. So me picking this pen up, for instance, my body recognizes that I've picked this up before and I understand the weight of it, and it's a rather small object. It understands that I don't need to use as many muscles to lift this and use this. Whereas if I try to lift this desk that my computer's on, then my body's going to say, okay, we need a lot more muscle force for that to occur. So your body and your brain, more specifically, has the ability to comprehend how many muscle fibers are needed to generate more force or less force. Now, in regards to this graded response down here, basically what this is saying is that if you have different degrees of skeletal muscle shortening or rapid stimuli, it can cause constant contraction or tetanus. The good news is most people don't have to worry about tetanus because of vaccination, but it's a pretty serious condition. It can cause your muscles to lock up, be very painful. It's like a permanent cramp. So here we're just kind of repeating what I saw on the last slide. Muscle force is dependent on the number of fibers stimulated. More fibers contracting results in greater muscle tension. What is the limiting factor? So, what is the one thing that can restrict your muscles? Energy. Is there enough ATP? If you run low on energy, then your muscles start to give out. So, how do we solve that? That's what we're going to learn more about in the next video. Okay. So, thanks for tuning in, guys. I appreciate it. I will see you on the next one.